Our next topic is our research update. We're very grateful to everyone for the donations and the activities that raise funds for support of our laboratory. The research program regarding the cause and management of Marfan was set up 20 years ago with a specifically equipped laboratory, courtesy of the family of Sonalee, a young doctor who died with Marfan syndrome. Our research director is Dr. José Aragon-Martin, and he's going to report on the research performed this summer by 4 summer students who were either self-funding or funded by the Girdlers Guild, to whom we are very grateful. Each student will give a brief report about their summer work. Dr. Martin has introduced this video, and it's recorded since he's in Mallorca at his brother's postponed wedding. So I present Dr. Aragon-Martin's recorded report.
Good afternoon. My name is Dr. Aragon-Martin, and I am the research director of Sonalee Laboratory since 2012. We are located at Guy's Cutting Building, Royal Brompton campus at Imperial College. Today, I'm going to talk about the research done during the summer in Sonalee Laboratory. And like the message says in the middle, things can only get better. But first, I'm going to talk about what areas Sonalee Lab covers. I'm also going to talk about funding Sonalee Lab research by donating towards consumables in the lab, then projects during the summer 2021, and at the end, I will talk about the possibility to come to Sonalee Lab as a volunteer or as a visitor. Let me tell you what area Sonalee Lab covers. The main pillar of research is Marfan syndrome. This work is done by myself and in collaboration with other groups. We are collaborating with Dr. Sanjay Sinha in Cambridge to discover new drugs for Marfan syndrome patients that will help to manage better the condition. Here at Sonalee Lab, we are looking at the genotype-phenotype association in Marfan syndrome because we want to find out if a particular mutation in the FBN1 gene produces a particular clinical feature, and in this way, try to predict the clinical features when a patient is being told for the first time about their mutation to assess better the number of times per year that the patient will require to come to the clinic for follow-ups. We are also working in the fine mapping of the FBN1 gene to describe better those variants that are causative and those that are just so-called normal. There are other areas of research that complement Marfan syndrome. We are looking to discover new genes in thoracic aortic aneurysm and dissection, ectopia lentis, scoliosis, and Ehlers-Danios. When a patient with Marfan syndrome clinical features comes to the clinic, it's our duty to find out the genetic cause in the patient. If the condition is fatal, like Marfan syndrome, it needs our utmost attention to find out if we can completely rule out FBN1 gene involvement. If we screen the patient in NGS, we will have more power to find the cause of the genetic condition. We can look at the variant position and also study the intronic regions of the gene. If we still do not find the cause, it might be due to large deletions in the gene or other chromosomal abnormalities that NGS cannot detect on its own in one patient. And here is the vital approach. Because we have NGS data on the patient, we can go back to this data and find out if there is another gene involved. In the case of Marfan syndrome patients with ectopia lentis, we can look at ADAN-TSL4 gene. And if with thoracic aortic aneurysm, we can look at TAD gene panels. There are about 63 genes in the TAD gene panels. It is imperative that we use our expertise in confirming or ruling out FBN1 gene. We know that causative mutations in ADAN-TSL4 gene have not been proved yet to be involved with cardiovascular clinical features seen in Marfan syndrome. Thanks to the fundraisers, Marfan Trust was able to donate money to research projects in Sonalee Lab during the summer. We hosted one Marfan Trust sponsorship student, 2 MSc projects, and 1 Europe student. Sonalee Lab wants to say thank you to all the students that helped us during the summer 2021. Husnaa was one of the Sonalee Lab Marfan Trust sponsorship students. She has been working finding current information about genotype-phenotype associations in Marfan syndrome, and here she is explaining her experience during the 8 weeks with us.
Hello, my name is Husnaa Fathima Khan, and I'm one of the fourth year medical students studying here at Queen's University, Belfast. So before I begin, I'd like to thank Dr. Antonio and the entire Marfan Trust team for giving me this opportunity to work on the project that is the genotype-phenotype associations in Marfan syndrome this summer remotely from Dubai. So briefly, just introducing the topic. So what is Marfan syndrome? So Marfan syndrome is basically like a connective tissue disorder, mainly autosomal dominant inheritance that is characterized by the loss of elastic tissue that results in affecting like various organ systems in the body that include like the cardiovascular, the respiratory, musculoskeletal, and much more. And typically, Marfan syndrome is caused by the mutation in this particular gene called the fibrillin gene, also known as the FBN1 gene. And in this project, not only did we focus on the position of the amino acid that has been exchanged in the process, but also the various type of amino acids that have been exchanged, resulting in the clinical features that are seen in such patients. So, you know, like being a fourth year medical student studying here at Queen's, we have clinical rotations throughout the entire academic year. And working on this project made me realize the undue importance of medical research and how it contributes towards the medical field. And working with Dr. Antonio again throughout these 8 weeks made me realize that I had hands-on experience, which fostered like my analytical skills and critical thinking as well, and made me focus on this very important topic that not a lot of people are aware about. So again, like I'd like to thank the entire team for giving me this opportunity again, coming here and speaking about this, and hope you have a good day. Thank you.
Husnaa research in the literature using PubMed website, information about the relationship between the FBN1 gene and the clinical features linked to a particular mutation. This work is very intensive and extensive, and so far, only the neonatal Marfan region has been seen as the only clear region. The difficulty of the neonatal region is that it also carries classical Marfan mutations. As far as we know, no one has attempted yet to establish a more clear understanding at a molecular level of a mutation and its clinical features or features. Dear Husnaa, thank you for spending time with us. We also have extra help in the laboratory from medical student Rohan, who during the summer was organizing the samples in the freezers. This will help us a lot when the Human Tissue Authority come for a visit. Here, Rohan is explaining his experience working with us.
We're here to meet Rohan, our Imperial College medical student who has very kindly volunteered his services to the Marfan Trust over the summer. He is sitting here in our Sonalee laboratory. Follow me. Rohan.
Hello.
Hello.
Hi.
Hello. You very kindly volunteered your services to the Marfan Trust this summer, and I thought you could just introduce yourself because I understand you have a history with the Marfan Trust that I did not know about. Okay.
So I'm Rohan. I'm going to second year medical student at Imperial College London, and I do have a bit of history of Marfan Trust. So I have actually did some work experience with José over here when I was in year 12, and I really enjoyed it then. I learned quite a lot of stuff about different genetic techniques and the work José was doing with Marfan patients, and I also had quite a strong interest throughout my A-levels with genetics. I did my EPQ and CRISPR, and when I then went to university, that continued, and I did have some teaching as well in genetics, but I felt that the kind of depth that I wanted was still lacking because I think it's a slightly neglected area of what we have been taught. And then I also found out that Jose had actually moved to Imperial. So once I found this out, I contacted Jose, and he said that I could very, very kindly, because actually it's been very difficult because I'm quite interested in research and I've not been able to find anything this summer at all. So Jose very kindly offered me to come back to the Sonalee lab for me to actually stay even longer and actually volunteer and do some proper work. So what I've been doing is sort of organising and reassigning the samples so that they can be used for future research. Like they have like lots of connective tissue disorders, like especially Marfan syndrome. So what I'm doing is sort of organising so that that can be used for future research. So yeah.
And what did the samples actually involve? I understand they're stored in the freezer,
Yeah.
and they came from St George's.
Yeah.
I knew that are precious samples. What did they involve? Who did they actually belong to? Who were they taken
So
from?
they belong to patients who have problems associated with connective tissue disorders like Marfan syndrome, and to screen them for particular genes that cause that. So, for example, some have scoliosis, thoracic aneurysm, and glaucoma. So all these, which are all connected to, which are all like complications of connective tissue disorders. And I think probably by me doing this, it's not just for myself; it's also for like helping Marfan Trust.
Rohan has also learned how to write family genetic pedigrees using the command line tool called Medline2. Genetic family pedigrees are very helpful when writing grant applications and establishing research projects. At present, Rohan has taken over Husnaa's project to continue with the Genotype-Phenotype Association, which is a major project, and it will take time to come with an answer. Dear Rohan, thank you for continuing helping us in the lab. MSc students Qasim and Lauren has been working in 2 different cohorts of patients with thoracic aortic aneurysms and dissections as the main clinical feature to rule out TAD genes. Here, Qasim and Lauren are explaining their experience working with us.
Jose, our laboratory director and scientist, is hosting 2 Imperial College MSc students, Lauren and Qasim, who have been sitting in this office in largely rapt silence since the beginning of June. Let's go and meet them. Lauren and Qasim, in no particular order, tell our audience what it is you're studying at Imperial College and what drew you to the Marfan Trust for the summer.
So I'm doing my Masters in Genomic Medicine at Imperial, and that involves all different sort of aspects of genomics in healthcare. The reason why I was drawn to this particular project and working here at the Marfan's Trust was because I'm quite interested in clinical genetics, and while looking for projects to do for my Masters, this one really interested me because it involves working with a rare inherited disorder. I'm looking at thoracic aortic aneurysm and dissection, which is an overlapping feature with Marfan's, and I'm looking at a cohort of about 20 patients which are unresolved cases, which means we don't really know which gene is causing their condition. In this project, I'm looking at the genetic data to try to identify the potentially causing gene and hopefully giving an answer to the question of their diagnosis.
So I'm also studying genomic medicine at Imperial, and I was particularly drawn towards my project in Marfan Trust because it was an opportunity to learn a range of really fundamental processes to identify causative variants in a very deleterious rare inherited disease. And yeah, so it's been a very eye-opening experience so far, and I've learned a lot.
Thank you, Lauren and Qasim. We're very lucky to have you here at the Marfan Trust, and we look forward to reading your report in September, which will be a big feature in our autumn newsletter. Thank you.
Thanks.
Thank you, Jose, cameraman, scientist, laboratory director. You can stop.
Both of them have a group of 20 TAD samples, and they both use NGS data to look for candidate variants. These variants need to be processed for interpretation using several computer tools. Qasim was able to find a variant in the FM1 gene that it was classified as causative, since it will shorten the protein length. And this type of DNA effect has been seen in many Marfan patients. This variant needs to be confirmed in another technique called Sanger sequencing, and other family members need to be tested to establish the genetic of the variant in the family. It is interesting that previous techniques did not pick it up, and it shows the strength of using NGS as a technology for DNA testing. Dear Qasim and Lauren, thank you for helping us in the lab. I would like to thank all the patients and their families involved in Sonalee lab research. Also, let's not forget the volunteers and fundraisers that always give their free time for this cause. To the Marfan Trust team that is always trying to help the Sonalee laboratory doing their best, students from all years, medical, PhDs, MScs, and BScs, who come to our laboratory to be trained, gain experience and knowledge about Marfan syndrome. And finally, the sponsors, which sometimes gives that extra help to push our research a little bit further. Now, I will like to approach to those who are interested in volunteering or visiting the lab. Are you a medical student? Is genetics part of your undergraduate degree? You can volunteer in the lab during the summer or through the year. We can train you in laboratory skills that will help you in your career. These are some of the students who have volunteered in the past and at present. Perhaps you are interested in visiting the lab because you are interested in science or just want to know face-to-face what we do with the funding that you kindly raise. This will also give you the opportunity to ask the question that you were not able to do before in a more informal way. These are the pictures of previous fundraisers visiting the Sonalee lab. If you would like to be a Sonalee lab volunteer or visit the lab, please contact the Marfan Trust on the following email: info@marfantrust.org. Sonalee lab wants to create a place where kids are inspired, so when they are ready to come to the lab, we can help them to improve their lab skills. If there are any questions to Sonalee lab, please send them by email to jaragon.m@ic.ac.uk. Thank you for inviting me to today's Marfan Trust Information Day. See you later from Sonalee lab.
Thank you, Jose. Have a good wedding in Mallorca. When he comes back, we have to move our lab, and this is a big undertaking. But we've made such strides over the years. In 1991, the gene was discovered by our international consortium of 8 countries working together. And it was one of the first to be put on the list for service screening by the NHS. It really has revolutionized our care. We can make a diagnosis in 97% of cases. And of course, it helps us to screen the rest of the family and say to people either, well, you're carrying the gene, we need to keep an eye on you, or you're not carrying the gene, we never need to see you again. So it's been a tremendous leap forward, this analysis of mutations. Are there any questions from the audience, Victoria?
MT would like to know the destination of the laboratory where we are off to.
That's a very good question. It's still under negotiation. We're trying very hard to stay at the Brompton Hospital, and we've had an offer. Next week, we have to go and see if the lab is big enough. But as Mr. Rosendahl pointed out, it really is a major center of excellence for Marfan syndrome. And we have the Southeast NHS genetic screening lab, which looks at all patients who have an aortic aneurysm and screens them for about 60 genes that are known around the world. The Marfan gene was the first one, but our 8 groups are still working together, and we're up to about 60 genes now. Some are unique just for one particular family. So we want to continue this work. We think there are another 70 genes to discover, and each of them then leads us to appropriate medical treatment, and we know roughly when to operate. So we're hoping to move into the Brompton in the coming year and pick up our research project to continue mapping the Marfan gene. There are over 2,000 mutations recorded worldwide, and to find new genes. So you should find us at the Brompton Hospital.